Hello, I'm Alan Handyside, Honorary Professor of Reproductive Genetics at the School of Biosciences at the University of Kent in Canterbury in the United Kingdom. It is a pleasure and an honor to present this lecture at this meeting of the Japanese Society of Human Genetics, and I'd like to thank the organizers for inviting me. It is sad that I cannot be with you in person because of the ongoing pandemic, but I look forward to the opportunity of visiting Japan again soon to meet friends and colleagues. July 2020 marked the 30th anniversary of the first births in 1990, following IVF preimplantation embryo biopsy and single-cell genetic analysis, which was then known as preimplantation genetic diagnosis, or PGD, and has recently been renamed preimplantation genetic testing. In my presentation today, as well as celebrating that early achievement, I will look back over the last 30 years and highlight just a few of the many scientific, technical, and clinical developments that have revolutionized our ability to test for an expanding range of monogenic diseases, chromosomal abnormalities, and other conditions at this early stage of human development. So first, my disclosure slide. I'm going to start today with a brief historical review and attempt to place some of the major milestones over the last 30 years in the context of advances in molecular methods and human genetics, before focusing on the development of preimplantation genetic testing for monogenic disease and preimplantation genetic testing for aneuploidy, as these are the two main applications of PGT. I will then summarize the benefits of incorporating PGT-A for embryo selection in IVF practice, and finally highlight some promising work that may indicate future trends in the area of preimplantation testing. Although Richard Gardner and Bob Edwards, the IVF pioneer and Nobel laureate, had successfully biopsied rabbit blastocysts to identify their gender and suggested similar methods might be used for testing human embryos to avoid X-linked conditions, in a Nature article in 1968, it was only almost 20 years later, with the development of human IVF itself and the first publication of the polymerase chain reaction, showing sensitivity down to about 100 cells in the mid-1980s, that genetic testing of human embryos could be seriously proposed. Looking back, the scale of the challenge is clear. Human genetics was still in its infancy, and far more was known about the genetics of model species such as the mouse. The genes involved in many common monogenic diseases were still unknown, and the first publications of the causative mutations, notably for Huntington's disease and cystic fibrosis, were just beginning to appear. Furthermore, it would not be for another 15 years that with progress in next-generation sequencing methods, that the first draft of the human genome was sequenced. And only as recently as 2014, that the cost of sequencing an individual's genome has been possible for $1,000, opening up a new era of personalized genomics. The main driver for developing preimplantation genetic testing in the late 1980s was to allow couples at risk of inherited disease to start their pregnancy knowing that it would not be affected, as an alternative to conventional prenatal diagnosis at a later stage of an established pregnancy, with the inherent risk, of course, of having to consider terminating an affected pregnancy. Initially, there was skepticism because of the low success rates following IVF, particularly at that time. The burden of the treatment for mainly fertile couples, which of course involves down-regulation, superovulation and egg collection, and so on, and of course the cost. However, following the publicity surrounding the first successful clinical application in births, it soon became clear that many couples considered this a valuable alternative and approached this for help. To perform genetic analysis, one or more cells needs to be removed or biopsied from each fertilized embryo. The Chicago group, led by Yuri Valinsky and Anne Vakuliev, pioneered polar body biopsy as an approach to diagnose maternally inherited conditions, and that's shown on the top panel on the right here. Also, at that time, Bob Edwards strongly advocated blastocyst biopsy because multiple trabectodome cells, which would not contribute to the fetus, could be sampled, potentially increasing the accuracy of the diagnosis. And that's shown here in the bottom panel on the right. However, although embryos could be cultured to the blastocyst stage, in the media we used them, pregnancy rates following transfer were low. So we chose to biopsy single cells from cleavage stage embryos at the 6 to 10 cell stage early on the third day following insemination, and that's shown here in the middle panel. But as we could not cryopreserve the biopsied embryos, we then only had 12 hours to perform the genetic analysis on the same day and to transfer unaffected embryos. Today, with improvements in culture and pregnancy outcomes, together with minimally damaging cryopreservation of blastocyst stage embryos by rapid cooling or vitrification, has transformed pre-implantation genetic testing, removing the time limit for genetic analysis. Most PGT, therefore, now involves blastocyst biopsy, vitrification, and frozen embryo transfers in the latest cycle. Okay, so I'm now going to focus in more detail on developments in pre-implantation genetic testing, in particular for monogenic disorders, or PGT-M, as it's now known. The first births following IVF and pre-implantation genetic testing were in a series of couples at risk of X-linked disease, which typically affect males only, by a small team led by myself and Professor Lord Robert Winston at Hammersmith Hospital in London. At that time, we'd not succeeded in amplifying unique sequences from single cells using the polymerase chain reaction, even with high numbers of cycles. But we were able to amplify a Y-specific repeat sequence that allowed us to identify male embryos, which is shown on the left here. When we began clinical application, we were concerned that the holes that we drilled in the protective zona pellucida would allow the embryos to escape and be destroyed in the uterus. However, in the very first cycle, the patient had a positive pregnancy test. Unfortunately, though, the pregnancy was not sustained. But by the autumn of 1989, 2 twin pregnancies were ongoing, which were subsequently born in July 1990. And this slide in the middle shows the embryos that were transferred at cleavage stages on day 3. And on the right are Natalie Danielle, at the top of the slide, the first 2 twins to be born. And then in the lower slide, myself, Robert Winston, and the parents of Rachel, who was the surviving child from the second set of twins, again born in July 1990, but this photograph taken in 1993. Although identifying the gender of embryos was applicable generally to the several hundred X-linked conditions which were then known, Clearly, only half of the male embryos not selected for transfer would have been affected. It was therefore clear that it would be preferable to identify the causative mutations in monogenic diseases themselves. The first PGT for monogenic disease in which the mutation was detected directly was in a couple at risk of cystic fibrosis, reported 2 years later in 1992 by our group at Hammersmith Hospital in collaboration with Mark Hughes' lab at Baylor College in Houston in the US. The breakthrough was to use nested PCR, that is 2 rounds of PCR with a nested pair of oligonucleotide primers in the second round. We then used heteroduplex formation and rapid mini-gel analysis to detect the common delta F508 3 base pair deletion causing cystic fibrosis, as we were limited still to day 3 transfers. This nested PCR approach was subsequently used for a range of other monogenic diseases, but was compromised by allele dropout, in which at the single cell level at least, there's a failure to amplify both of the parental copies of the fragment, and also contamination, particularly the buildup of product contamination within the lab, which had the potential to cause misdiagnoses. With the development of fluorescent PCR and fragment analysis by capillary electrophoresis, together with the identification of highly polymorphic short tandem repeats across the genome, multiplex mini-sequencing for the mutation, and targeted haplotyping by nested fluorescent PCR and analysis of flanking STR markers were developed for cystic fibrosis and a series of other monogenic diseases to avoid the previous problems of allele dropout and contamination and increase the accuracy. And indeed, this approach would become the gold standard for the next 15 years. One major achievement over this period of time was preimplantation genetic testing for a series of different monogenic diseases combined with HLA matching. And this was first reported by Walensky's group in a case of Fanconi's anemia, and has since then been applied to several hundred similar blood-borne conditions, including things like beta thalassemia, for example, allowing unaffected cord blood stem cells to existing affected children within the families, and has cured many of them to date. The major drawback of this approach, however, was that it required patient and disease-specific protocols to be developed, which limited the availability of PGT to specialized labs and clinics and delayed treatment for many patients. The next major breakthrough was the discovery of a heat-stable DNA polymerase from the bacteriophage 529 with topoisomerase and proofreading capability that allows isothermal multiple displacement amplification, or MDA, in the early 2000s. This meant that for the first time, the whole genome of picogram quantities of DNA present in single or small numbers of cells could be amplified to generate micrograms of DNA and open up the possibility of analysis on genome-wide platforms. This had a revolutionary effect, not only for PGT of monogenic disease, but as we'll see later, for PGT of chromosomal aneuploidy. For PGTM, genome-wide SNP genotyping by beta-array or next-generation sequencing and haplotyping of parental chromosomes by carrier mapping and other algorithms now provides a universal set of markers for linkage-based testing of virtually any monogenic disease, and can be combined with molecular cytogenetics. The most important thing about this approach is that it avoids the need for any patient and disease-specific workup and has made pre-implantation genetic testing much more widely available. Here I show one example of beta thalassemia showing that the parental haplotyping shown for the paternal chromosome in red and blue and the maternal chromosome in orange and green allows the accurate identification of the position of crossovers in embryos to be detected with high resolution. And comparison with a relative of known disease status of the disease locus, beta globin in this case, then allows the status of each embryo to be very straightforwardly determined. As the genome-wide SNP analysis provides hundreds of markers across each chromosome, the same test allows meiotic trisomies, monosomies, and deletions to be detected. In the example shown here, carrier mapping has been used to identify an unbalanced embryo in a couple where the mother is the carrier of a reciprocal translocation between chromosomes 11 and 22. So this clearly shows for chromosome 11 the absence of SNP markers at the distal end of the Q arm of chromosome 11. And because of the rapid transition between the haplotypes on chromosome 22, the presence of an additional chromosome, maternal chromosome segment at that point for chromosome 22. But unlike copy number analysis, by examining the haplotypes at the translocation breakpoints, it is possible to distinguish normal and carrier embryos. And many couples would prefer to select normal embryos for transfer if they are available, so that any resulting child will not suffer the same infertility or repeated miscarriages as the parent had. The first births using carrier mapping were in 2015 and 16 in the United Kingdom for Charcot-Marie-Tooth disease, in Australia for congenital myasthenic syndrome and breast cancer predisposition, and in China for congenital adrenal hyperplasia. The second major application for pre-implantation genetic testing is for chromosomal aneuploidy, or PGTA. And I'm now going to discuss how this field has developed over the same period of time. I'm going to start with IVF success rates as they are today by showing the Japan registry data for 2017, which was recently published, and provides a very valuable benchmark for fresh and frozen single embryo transfers, particularly at the blastocyst stage, since Japan pioneered the technique of vitrification. Live birth rates have improved significantly since the late 1980s and 1990s, with rates of about 35% per cycle with an embryo transfer in women below the age of 35 years, even with single embryo transfers. However, for women in their late 30s and 40s, live birth rates rapidly decline, as shown in the yellow line here. And at the same time, miscarriage rates increase exponentially until by the age of 45, the chance of a miscarriage is above 50%. So the rationale for testing for aneuploidy is very clear. And it's now been well established using a variety of techniques that the incidence of chromosomal aneuploidy following IVF is very high and is a major cause of IVF failure, therefore, pregnancy loss, abnormal pregnancies and affected live births, and increases in human eggs exponentially with maternal age above the age of 35. Furthermore, we know that the incidence of recognized aneuploidies at birth is only between 0.3 and 0.5%. So a natural selection occurs through, first of all, pregnancy failure, pregnancy loss, and so on throughout pregnancy. And so it's clear that, if possible, we should avoid transferring these aneuploid embryos. PGT by next generation sequencing now allows accurate copy number analysis for all 24 chromosomes at high resolution. And it's being used widely as a highly effective additional tool for embryo selection in combination with blastocyst culture, time-lapse or morphology scoring to ensure that the best embryo, if there are several euploid embryos, the best embryo is transferred, biopsy and vitrification, and particularly for those programs focusing on single blastocyst transfers. Even in the mid-1980s, when live birth rates were much lower than they are today, it was suspected that chromosome aneuploidy might be one of the reasons, and cytogeneticists like Rosangel published data showing a high incidence of a range of chromosomal abnormalities. However, karyotyping chromosomes in human eggs and embryos is technically very challenging, and banding to identify individual chromosomes was not possible. It was only in the early 1990s, with the development of multicolor fluorescence in situ hybridization to interphase nuclei, generally with probes to 5 to 7 chromosomes, mainly the small acrocentrics associated with viable but abnormal pregnancies, that the high incidence of aneuploidies of meiotic and mitotic origin was demonstrated in cleavage stage embryos. This led to the first attempts and the first births following PGT, following these probes, the use of these probe sets in 1997 by Giannaroli and colleagues, including Santiago Monet. And this became widely practiced. Although this undoubtedly helped some women having IVF avoid pregnancies affected by these viable trisomies and monosomies, randomized controlled trials showed that pregnancy and live birth rates were not significantly improved, and it was realized that the limited number of chromosomes which could be analyzed routinely by multicolor FISH was one of the main limitations. However, it would be another 10 years before 24 chromosome copy number analysis was finally possible using whole genome amplification using a PCR library-based method and array comparative hybridization. The first birth was announced, in fact, following first polar body analysis and array CGH in September 2009. And here we just see a nice photograph of Simon Fischel in the bottom right, holding Oliver, which was the first child to be tested in this way. In this particular couple, they'd had 13 previous failed IVF cycles, and Simon used first polar bodies for the analysis only. We now know that it would be more accurate to analyze both the first and second polar bodies, and in this case discovered that 7 out of 9 of them were aneuploid, having transferred the 2 that didn't have an aneuploid in the first polar body, Oliver they succeeded, and Oliver was born later. Then in 2014, next-generation sequencing-based copy number analysis became available by low read-depth sequencing and mapped fragment counting. So this technique involves sequencing 36 nucleotide fragments, mapping them back to the reference human genome, and binning the numbers of fragments which map to small regions across each chromosome allows accurate copy number analysis to be performed. This has the advantage that copy number is the relationship between copy number and the original sample following whole genome amplification is linearly related to the numbers of fragments that map to the individual chromosomes. And also because we can do this across many regions of each chromosome, we can do high resolution analysis. In this example, you can see here a clear trisomy for chromosome 8 with 3 copies and a monosomy for 21 in this trabectodon biopsy sample. One of the pioneers of PGTA using array-CGH and then NGS-based testing is Robert Anderson and his group in Southern California in the United States. And they've recently published their experience using PGTA for all patients, irrespective of age or prognosis, over a 6-year period from 2012 to 2018. Here I've replotted their outcome data and compared it with the 2017 Japan registry data in yellow. And this group, I should emphasize, is also uses predominantly single blastocyst transfers, so the data is comparable. What we can see is that irrespective of maternal age, the implantation rates which they achieved, shown in the blue bars, are really very high, much higher than the implantation rate, the corresponding implantation rate in the registry data. And they're maintained irrespective of the patient's age, all the way up through to the early 40s. And in fact, the average implantation rate for embryo transfer, of course, was 75%. So this is incredibly high in relation to normal practice of IVF without pre-implantation genetic testing. However, of course, it's well known that as women age, IVF is less successful. There are fewer eggs and embryos, so that in some cycles there are no embryos which reach the blastocyst stage, or if they do reach the blastocyst stage, there are fewer of them. And as you get into women in their 40s, then the possibility that all of those embryos are aneuploid and not available for transfer increases. So this group calculated the live birth rate per cycle, and in the younger age groups, of course, the rates remained really exceedingly high. So up to the age of 40, it exceeded 60%. As you come into women in their 40s, of course, it becomes more difficult to find a euploid blastocyst for transfer, and the rates come down to the rates that are more comparable with what is seen in the Japan registry data. But nevertheless, the very striking increase in the implantation and live birth rates per cycle, at least in women up to the age of 40. Another recent study, just published online, dramatically demonstrates the clinical outcomes of transferring single euploid versus single aneuploid blastocysts. To do this, they performed a non-selection study. In a non-selection study, patients are consented for single blastocyst transfer following biopsy, but the PGTA results are only unblinded after the clinical outcome in each case is known. Performing this analysis in this study, they showed that 60% of the blastocysts were euploid, about a quarter of the blastocysts were aneuploid, and the remainder either had mosaic or segmental copy number abnormalities, and in only 2% did they fail to get a result following trophectomy biopsy. So what you can see on the right is that for euploid single blastocyst transfers, as shown in the blue, the positive HCG rate or implantation rate was exceeded 80%, and this is in all women treated of all ages. The clinical pregnancy rate, as shown by the fetal heart rate, decreases to some extent. There are some losses, but the ongoing pregnancy rate and delivery rate was actually about 65%. Considering that this is the average rate, that's really very high, showing the power of selectively transferring euploid blastocysts. On the other hand, of those embryos which were transferred and shown to be aneuploid, the implantation rate was less than half, at around 40%. There were significant losses before the fetal heart stage in terms of clinical pregnancy rate, and none of those pregnancies were ongoing or resulted in a live birth. So this really shows that we should be avoiding the transfer of aneuploid embryos, if at all possible, because the only outcome, of course, is the likelihood of a miscarriage at some stage. Furthermore, they compared the ongoing pregnancy rate and delivery rate of non-biopsied blastocysts in a retrospective comparison group with all of the biopsied blastocysts, irrespective of the diagnosis. And in those cases, they showed that the ongoing pregnancy and delivery rate for non-biopsied blastocysts was about 46%, and for the biopsied blastocysts, 48%, which was clearly not significant. And so removing cells by trophectoderm biopsy at the blastocyst stage does not appear in their hands to cause any detrimental effect in terms of clinical outcome. So the benefits of PGTA then for IVF practice, of course, are the high implantation and clinical pregnancy rates and low miscarriage rates with single euploid vitrified warm blastocyst transfers. But it also means that patients can cryostore euploid blastocysts for future transfers and essentially have their family in the freezer, rather than storing large numbers of embryos, many of which may be aneuploid and essentially not viable. Of course, the testing also reduces the risk of chromosomally abnormal pregnancy and live birth. But one of the main advantages is that it improves clinical management for these patients and embryology standards. Because implantation and pregnancy rates are so high, it gives you a very accurate key performance indicator for anything in the clinical and embryology protocols that perhaps is not optimal. And finally, of course, it enables patients to make informed decisions about the likelihood that they can have a baby with their own eggs and embryos. And that will allow them to make informed decisions. And of course, if an older patient wishes to continue with IVF, then that is her choice. but otherwise then perhaps they can think about oocyte donation. So finally, future trends in pre-implantation genetic testing. Well, the most exciting development in this area is the recent discovery that there is cell-free DNA in the spent culture medium and/or blastocyst fluid of embryonic origin. So there's intense interest in whether we can use this as a source of DNA for testing, particularly for aneuploidy. And there have been a variety of studies, and it's now reached the stage where some groups are actually using it clinically. And of course, this has the huge benefit that instead of an invasive biopsy, which is labor intensive and skilled, non-invasive collection of the spent culture medium can be used. And that should make pre-implantation genetic testing more broadly available. Whether or not the accuracy of the diagnosis will allow PGT for single gene defects is not clear at this stage. However, there's also synergy with other recent studies showing improvements in IVF generally, because one thing that has become clear over the last 30 years is that success following pre-implantation genetic testing is really down to the numbers of human eggs and embryos which are available in the cycle to test. So recent exciting developments include, for example, this report from Furuhashi et al. Showing that piezo-ICSI, that is to say, electrically controlled intracytoplasmic sperm injection, can actually increase fertilization rates and the number of blastocysts, particularly in women above the age of 35 years, when we're desperately trying to identify that single euploid blastocyst, which we know will have a good chance of implanting and developing. Furthermore, there's also another recent study that shows improved embryo development, similarly, particularly in older women, using time-lapse incubators and low oxygen tensions, for example, because these incubators mean that the embryos are not disturbed during their culture. And again, if you put these techniques together and we can improve the numbers of eggs and embryos, then of course that's going to be a tremendous benefit. And finally, of course, if you are culturing embryos in a time-lapse incubator, you can use time-lapse analysis to look at the development so that you can, if you're lucky enough to have a number of euploid embryos, you can improve selection and optimize and select the best embryo for transfer. And recently, people have been looking at machine learning and artificial intelligence to do this. And I think there's going to be some very interesting and exciting developments in this area. So in conclusion, the last 30 years has seen major technical advances in single-cell genetics and genomics, allowing the efficient and accurate pre-implantation genetic testing for an expanding range of conditions. And our knowledge of human genetics has also been transformed by the sequencing of the human genome. One group in the US is even now using genome-wide SNP analysis for polygenic risk scoring of embryos for common diseases such as diabetes. So I believe we have a lot to look forward to over the coming years. Thank you for listening.